perfect. All right. So let's keep going. So in photosynthesis, the main pigment responsible is actually chlorophyll A. So chlorophyll A can basically use the light energy and then transfer that energy and bounce up the energy level of some electrons. But there are other pigments which are also called accessory pigments. So they can actually absorb different wavelengths of light energy. And then examples of these are chlorophyll B, for example, and also carotene. And xanthophyll is actually another one. So it's a yellow colored pigment. So all of them can capture some energy that chlorophyll A cannot, because each pigment has different property and they have different absorption spectrum. But they cannot convert that energy directly to elevate an electron. They have to pass on that energy to chlorophyll A, and only chlorophyll A can basically use the energy to bounce up electron energy level. So that's why these are called accessory pigments, and chlorophyll A is the main photosynthetic pigment. So what is the absorption spectrum? So that's literally the wavelengths a particular pigment can absorb. So the way you measure that is basically if you have a solution of chlorophyll A, for example, you put it into a cuvette. So you actually use a spectrophotometer. You put it into a cuvette, and then you set the spec at a particular wavelength. So let's say you start at 350 nanometer, and then you measure the wavelengths. So the y-axis over here is basically the absorption. So the spec will spit out a number. And then you change the wavelengths to 370, and then you measure the absorption again. And then you basically, it's kind of tedious. You go all the way to more than 700. And then at some wavelengths, you're gonna see a peak. So that means that particular pigment, so this is actually chlorophyll A, can absorb a lot of the light within 400 and 450. And then in the green area, there's nothing because they cannot absorb those light. But then once you get into the red area, so between 650 and 700, chlorophyll A can also absorb quite a bit of light. Chlorophyll B is kind of similar. If you look at the structure, it's kind of similar to chlorophyll A. It also, it's this dotted line over here. It also absorbed in the blue area, or mainly kind of bluish area, and then in the reddish area. But you can actually see that the peaks are not completely overlapping each other. So it can gather some light energy that chlorophyll A cannot. And then for carotene, the peak also shifted a little bit away from chlorophyll A. So that means that they can gather some light that chlorophyll A cannot. So if you look at light-dependent reactions, so it is happening on the thylakoid membrane. And there are actually two complexes called photosystem 2 and photosystem 1. It is a little bit confusing because they're named after which one is identified first, not after which one is doing their job in a plant photosynthesis. So in plant photosynthesis, it's actually photosystem 2 that's first, and then photosystem 1 that's second. And in both photosystem, light energy can actually be captured by chlorophyll, and then the chlorophyll can actually use that energy to basically increase the energy level of some electrons and then do some work. Okay. Right. So the question is, which component of the light-dependent reaction produce NADPH? So in light-dependent reaction, this is just another diagram. We end up producing both ATP and NADPH, and then of course oxygen. And remember, we talked about there are two photosystems. So the question is, which of these produce NADPH? The correct answer is actually photosystem 1. It's the second one. And let's see, who do we have on the leaderboard? We have Helen, Roop, Nikhil, Malika, and Charles on the leaderboard. So here's kind of a closer look of what the two photosystems do. So photosystem 2, remember it is the first one over here. And so basically it has these protein complex called the antenna complex. And the pigments like chlorophyll A and the other pigments are basically embedded in these protein complex. And so light hits, so some photon gets absorbed by the pigments, and the pigments, the chlorophyll A actually, so in the middle of the reaction center, there is actually a special pair of chlorophyll A. They literally are called the special pair. So they can elevate, they can use the energy from the photons and elevate the energy level of electrons. And then electron actually gets passed down through electron transport chain that's really, really similar to the mitochondria electron transfer chain. So bouncing from one complex to another. And then in the process, the cytochrome complex can actually use some of the energy to pump protons going from the stroma into the thylakoid space. So basically, inside the thylakoid lumen, there is an accumulation of proton, and then that is the set up a very steep proton motive force. And then when the proton flows back through the membrane, through the ATP synthase, we can actually use that concentration gradient to synthesize a whole bunch of ATP. And let's see. So Kate's asking, so plastoquinol over here, it's part of the electron transport chain. Yes, so it is part of the electron transport chain in chloroplast. And this is just another closer look of the antenna complex and the reaction center. So here is the special pair of chlorophyll A. And they basically bounce the energy level of the electron. And then the electron will get passed on to plastoquinol and then get passed on to cytochrome complex, which is a proton pump. So photosystem 2 is really setting up that proton pump so that we can actually make ATP. Photosystem 1, so this is the second one, is the one that actually produce NADPH. So the pigments can also absorb photons, and then they can also elevate energy level of electrons. And then electron actually gets passed to this next player, which is called ferredoxin. And then ferredoxin passes the electron to the next player, which is called FNR. So, for our purpose, for this class, you actually don't really need to remember the name of the enzyme complex, as long as you understand the concept that, electrons are being bounced from one party to the next, but then it's the FNR that's actually reducing NADP plus to NADPH. And that's actually happening on the stroma side of the thylakoid membrane. So remember, in the first one, we're pumping a lot of protons from the stroma into the thylakoid lumen. And then over here, that's actually another mechanism to reduce the proton concentration in the stroma side, because when you're reducing NADP plus, you're actually using some of the proton. So basically, the end result is you actually have a pretty steep proton gradient across the membrane. And this is just a visual to show you, so we actually didn't talk about it, but when it comes to redox reaction, there is this thing called redox potential. And it's a little bit too complicated to talk about, to go into it, but basically the more negative you are, the more redox potential you have. So you can actually see for both photosystem 2 and photosystem 1, the cell is capable of basically increase the redox potential of these electrons, way up. And then, going through electron transport chain, going from plastoquinone to these other the complex, and then plastocyanin, the energy level kind of goes down as the electron is bouncing along. But then once they get to photosystem 1, there's additional energy, and then the energy level goes up again. And then as they get past to ferredoxin and then FNR, the energy level is also going down. And then of course in photosystem 2, so the electron, where does electron come from? So for plants, that electron actually come from water. So there actually is an enzyme complex that's capable of splitting water. And then in the process, we are releasing some oxygen. Some proton are also being released, but the electron are basically being donated to the reaction center, and then end up being the high energy electron, and then going through the electron transport chain. So not all photosynthesis use water as the ultimate electron donor. So because of that, not all photosynthesis can actually produce oxygen. So the only reason plants photosynthesis end up releasing oxygen is because of this step of photosynthesis. Okay, and if you look at this, photosynthesis really is the reverse of cellular respiration, because in cellular respiration, oxygen is the final electron acceptor, and then oxygen gets reduced into water. And then over here, water is the initial electron donor, and then end up producing oxygen.
Just really quick while we're doing this, for curiosity's sake, why is photosystem 2 first? And like, why would they name it 2 and then that's the one that comes first and photosystem 1 is second? Like, is there a reason why they're called 1 and 2 when it seems like it should be the opposite?
1 was the first one to be discovered.
Okay, sorry.
And then after that, they discovered there is another photosystem, so they named the other one 2. But in terms of function, 2 actually happens first. Yeah.
Yeah. Okay. Just curious. Thank you.
Okay, much better this time. So we just talked about the light-dependent reaction, and it's happening on the thyroid membrane. This one is asking, so where does the light-independent part, which is the Calvin cycle, happen? And the correct answer is actually in the stroma. So if you look at this diagram, it's also kind of showing that, okay, so the Calvin cycle is actually happening in the stroma of chloroplast. So the Calvin cycle, just like Krebs cycle, is a cyclic pathway. And so like I said, photosynthesis really is kind of a reverse of cellular respiration, because in cellular respiration, we have Krebs cycle feeding into electron transport chain. And then in photosynthesis, we have electron transport chain, and then we feed into Calvin cycle. So it's a cyclic pathway. And we're actually not going to go into too much detail of the pathway, just to know that this is happening in the stroma of chloroplast. And the first enzyme is this enzyme called Rubisco. And it basically taking CO2 as inorganic carbon, and then incorporate it into a 3-carbon molecule, and eventually forming a 3-carbon molecule called glyceride triphosphate, or G3P. And then G3P is what's being released, and G3P can actually be used for synthesizing, glucose, and then other things in the cell. And then it is a cyclic cycle, so the rest of it, we're actually regenerating this compound, which is called RuBP. So this is the compound that goes into the cycle, and this is the compound that's being regenerated in the last step. And also the other thing to know is if you look at the cyclic pathway, all the ATPs were produced, that was being produced during light dependent reaction, are basically being used over here. So it's actually very energy intense. So you need 6 ATP over here, and then you need 3 ATP over here. The NADPH that was being produced in the light-dependent reaction, so you actually need 6 of them over here to basically donate their electron and produce G3P. So some of the product can actually stay in the chloroplast, and then other ones can actually be shuffled into so all of the chloroplasts go into the cytoplasm, and then there they can actually be used for synthesizing, sugar, fats, amino acid, and all kinds of. So, another similarity between this and Krebs cycle, it can actually produce intermediates for other pathways. So, in plants, if you look at it, so here's the chloroplast. This is where photosynthesis is happening, and the end result is sugar, and of course some oxygen is being released. So even though the light reaction end up producing some ATP, the ATP actually usually do not get out of the chloroplast, and that's partly because the inner membrane of chloroplast does not allow ATP and NADPH to actually get out. So they stay inside the thylakoid. So that's why actually for plants, most of the ATP they need for doing other function also come from cellular respiration. So the sugar that are produced from photosynthesis end up going to their mitochondria, and then they go through the same process of glycolysis, citric acid cycle, and then electron transport chain to produce ATP. So most of the ATP that are generated in the light dependent reaction are pretty much being used for making sugars. And that, of course, is it's great for not just the plants, it's great for the entire ecosystem, because if they don't make sugars, we won't have a way to get our energy. This is actually a little bit additional outside of the textbook, but it's kind of fun to talk about. So I'm just going to talk about this. So scientists actually think that this enzyme, so ATP synthase, is really important in cellular respiration, and it's really important in photosynthesis. So they actually think that initially it evolved from an ATP driven proton pump. So basically it's a membrane protein, and it uses energy from ATP hydrolysis to pump proton outside of the bacterial membrane. And so one of the things that we didn't mention about when we were talking about enzyme is actually a lot of the enzymes are actually capable of driving reactions in both directions. So it kind of just depends on sometimes, if the substrate is shifting, or depends on the demand of the cell, they can actually perform a different reaction. So this initial one is actually opposite of what we're seeing. Because what we're seeing over here is actually using the proton gradient to synthesize ATP. But they think that initially it's actually a proton pump that can pump proton outside of the cell and using energy produced by ATP hydrolysis. And then at some stage, there is another proton pump that evolved, and that's driven by electron transport chain. And then eventually these two things are being connected. And then so now we actually have these proton pumps that can pump hydrogen ion, and then the ATP synthase. The main function is actually synthesizing ATP using the proton gradient. However, if the condition in a cell changes, so if the cell has a lot of ATP, doesn't need to synthesize more ATP, the same enzyme can still do that. So it can still actually use energy released by ATP hydrolysis to actually pump hydrogen ion against the concentration gradient. Okay, so that's our record today. Photosynthesis convert light energy into chemical energy, and then of course that's what's driving the ecosystem. And then heterotrophs can actually eat the sugar, and then that's how we get our energy and stuff. Let's see, we have Helen, Rup, Nigel, Malika, and Charles, still the same 5 people. Okay, so the main objective for this lab is to kind of measure the absorption spectrum for each pigment. And so we are using a spectrophotometer. So it's an equipment that kind of look like that. And then you use a cuvette. And basically the solution that contain the pigment, you put it into the cuvette, it goes into the machine. And then so that's a knob that can adjust wavelengths. And then you basically show, you can measure the absorption and then use that to plot your absorption spectrum. So absorption are usually represented by A. So remember, this is the light that's being absorbed by the sample. And it is proportional to the concentration. So, if you have a chlorophyll A, solution in your cuvette, if it's a very low concentration, it's not going to absorb a lot of light. But if you have a lot more chlorophyll A, it's going to absorb more. And then the flip side of that is transmittance. So remember, transmittance is the light that gets transmitted after the cuvette. So the way to calculate transmittance is literally the light that has passed through divided by the light that's coming in. And so absorbance and transmittance actually are related. So they are actually have a log scale. It's actually a lot easier to talk about it with this slide. So here's your cuvette, and then your cuvette contains, this is the solution that contains your chlorophyll, for example. So the incident intensity light, so that's the light that's coming in, and then the transmitted light is basically the light passes through the cuvette, and then this is the light that comes out. So to calculate the percentage transmittance, it's basically this light out divided by the light in. And then it is a percentage. So if this cuvette contains, depends on what your solvent is. So if it's water, for example, or if it's an organic solvent like acetone, if it's just acetone, it's completely transparent. So your transmittance could actually be about 100%. But then once you have the pigment and some of the stuff, your transmittance is going to be somewhere between here. Until if it's really, really dark, it's not even going to transmit anything, then it could be 0. So absorbance is the flip side of it. So this is the equation to calculate absorbance. So it's actually minus log T. And absorbance, so the highest absorbance is actually 2. So that's basically minus log of 100. And then the lowest absorbance is 0. So this is if you have just the solvent, so acetone, it's not going to absorb a lot of light. And then if you have a lot of the pigments, it's going to absorb a lot. So that's the relationship between absorbance and transmittance. And as I mentioned earlier on, the way you measure absorption spectrum is you literally put the cuvette in there that contains one of the pigments, and then you will just go measure for every 20 nanometer what is the absorption, and you can actually see. So the absorption over here is 0 to you can set it to 1, or the highest one could be 2. And then try to figure out what is the peak. So that's the wavelengths that particular pigment will absorb a lot of energy. And so different pigments actually have different absorption spectrum. It's actually a lot easier to look at it over here. So here is chlorophyll A. You can see one peak in the blue region, and then another peak in the red region. Here is chlorophyll B. It's also one peak in the blue region, and then another one in the red region, but the peak are actually shifted a little bit different from chlorophyll A. And then carotenoids actually only has one peak, so it's more towards the green region over here. So that's absorption spectrum. There's this other thing called action spectrum. What is that? So action spectrum is basically what is the spectrum of light that plants or other organisms can use for photosynthesis. And it's literally the grand total of all these pigments kind of added together. So back in 1883, this guy, Thomas Engelmann, actually did a really clever experiment to measure the action spectrum. So the setup is actually really simple. So he used a prism to literally split visible light. So then you can actually see a rainbow going all the way from violet to red. And then the organism he used was this algae called Spirogera. So it's one of my favorite algae because they can actually arrange their chloroplast kind of in spiral. And then to measure the efficiency of photosynthesis, because remember photosynthesis produces oxygen. So he actually put these aerobic bacteria over there because these bacteria will only grow if there is a release of oxygen. And then he ended up producing an action spectrum kind of like that. So basically in the blue region, bacteria are very happily growing. In the red region, the bacteria are also very happily growing. But in green region, the bacteria are not growing because guess what? Spirogera, just like land plants, cannot absorb green pigment. And then they cannot use that energy spectrum for basically performing photosynthesis. So how come plants do not use green light for photosynthesis? There have been a few kind of arguments. One is that you have to balance between getting enough energy for photosynthesis and also overheating. So that particular argument says that if you actually absorb all the energy in the entire spectrum, you might actually end up overheating the plant. Another argument is actually blue light. So remember, they do have more energy. So absorbing that kind of more than make up for the loss of green, the green spectrum. But actually the best reason is actually for evolutionary reasons. So cyanobacteria, remember plants came from cyanobacteria. There are other bacteria called purple bacteria. So some of these are purple sulfur bacteria. Other ones are purple non-sulfur bacteria. These bacteria are actually using different pigments. So, things like carotenoids and stuff to perform photosynthesis. And for purple sulfur bacteria, so they actually use hydrogen sulfide, for example, instead of water as the electron donor. And then they're releasing, sulfur instead of oxygen. So they are actually absorbing green light for photosynthesis because they use different pigments. So that actually scientists speculate that cyanobacteria and purple bacteria end up co-evolving, and they end up using, basically they go into different ecological niche. So they absorb different spectrum of light so that they can actually coexist. So they can both perform photosynthesis, but if one of them absorb green light, the other one absorb everything other than green light, they can actually both coexist and survive together. Okay. Okay. Wow. This is really interesting distribution. We basically have people voted for all four answers, and they're fairly evenly distributed. The question is which pigment is not being used for photosynthesis. So if you look at the graph, remember chlorophyll A definitely is the main one. Both carotene and chlorophyll B are accessory pigments. Dentophil is also accessory pigment. The correct answer we're looking for is actually anthocyanin. So they're not usually involved in photosynthesis. They are involved in producing colorful colors. So they're, involving other features of the plant, such as reproduction and stuff like that. And then one other thing before and this is my last slide. So the reason I mentioned the purple bacteria, and they use keratin and other pigments, and they can absorb green light, is because, I assume most of you guys have done the pigment extraction simulation. So the premise of that simulation is actually you have just discovered this new green light absorbing pigment, and you're trying to use that, and then make biofuel and stuff like that. So, okay, so let's see who's on the leaderboard today. Oh, we have Susanna, Kylie, and Megan. All right, so completely different people. And then Will and Helen are also on the leaderboard. So, okay, so good job, guys. And let's see, before I send you guys to your breakout room, let me just open the homework. And then, so one other thing I want to show you guys is I have modified the syllabus. So remember, syllabus is in a Google Doc. I have changed the schedule just a little bit because early on, some of the lab are showing more than one simulations and stuff. And I have finally got some time to check out some of these simulations. So we're actually only gonna do mitosis next week, not both of these. And then we'll do meiosis in the following week, and then we'll do genetics in the following week. So make sure you guys download the updated syllabus because it reflect what most likely will be happening. It doesn't mean that I'm not gonna change any of these, but in general, this is a more updated version of which simulation we're actually gonna do. And before I send you guys to your breakout room, is there any other question?
I have a couple questions about the lab report.
Yeah.
So I was going through it and I was wondering, for when we're talking about the experiment, do we talk about the basketball and the mouse, or do we talk about
yes.
Okay, so we
Both.
could make that as one large experiment.
Right.
Okay, and
because
then
they're asking related questions, right? So you're just using different models. Yeah.
Okay. And then do we need to talk about how we had those simulations where we learn about the Krebs cycle and stuff?
Do you need to talk about
do we have to talk about that as part of our process at all, or can we just use that information in talking about the the topic?
That's a tough one. Does anybody else has any idea about that? Because I can see it both ways. I can see it as just background information, and then just assume when you're writing a paper, assume all your audience already know about Krebs cycle, or you can put a little bit more detail. I would leave that to you guys and see if you have a preference or not. I don't think the rubric says either way.
Okay. Yeah, I was originally thinking that I would use it, to talk about the concepts that we were learning about, but I wasn't going to be during my experiment, I filled out this sheet on the wall, that sort of thing.
Yeah, so I will leave it for you guys to decide if you want to include that much detail or not.
Okay.
But either way, I don't think I'm gonna use point for that. Just that alone.
Okay.
Yeah.
Thank you.
Okay.
Yeah.
Any other questions before I send you guys to your breakout room? No. Okay. So this past week, I did assign 2 simulations. One is required, the other one is more for extra credit. I had the modified syllabus. I'm trying to avoid that because I understand everybody has work and stuff. You do have other obligations, and you probably don't have a lot of time. But there might be one week where I might assign 2 simulation. And I'm wondering what you guys think about that. So when you do your daily reflection today, do you have strong opinion about it and not? So go ahead and put that down and just let me know. Okay, and your breakout rooms are open.